Right, so we have a talk from Professor Fermi here today, and Aart graduated from the Physics and Astronomy department at the Free University of Amsterdam in the Netherlands, and then he moved to Munich, and he received his PhD degree in experimental physics from LMU. Ludwig Maximilians University in Munich, which is the #1 university in Germany. And the title of his research, the subject of his research was absolute phase of few cycle and attosecond pulses to control the atomic scale motion of electrons. And of course, as you all know, the latest Nobel Prize in Physics in 2023 has been awarded for the achievements in attosecond science, specifically for the methods that generate attosecond pulses. So this is the citation, the Nobel Prize citation. So Aart has been working in this field already in 2006, 18 years ago, maybe 2006 is when Aart was moving to Vienna, but it was over 20 years ago. So he has been working in the field of attosecond science, and he was on some greatest papers on attosecond physics, including one of his papers from that time, attosecond real-time observation of electron tunneling in atoms, 2007 Nature paper. I checked it this morning. It has been cited more than 1,100 times as of this morning. And his other work on the control of electron localization in molecular dissociation has been cited almost 1,000 times. And his thesis supervisor during his time at Max Planck Institute in Garching and the LMU in Munich was Ferenc Krauss, who was one of the Nobel Prize winners this past fall. Following his work in Munich and Garching, Aart has moved to Vienna, where he contributed to the development of some unique laser sources, continuing to contribute to the field of attosecond science. But he also started his journey towards multiphoton microscopy, nonlinear microscopy, and biophotonics. And as a culmination of this journey, and as the highest point of this journey, Aart has ended up here at Texas A&M University, and he is our ambassador. Let me describe it this way: the ambassador of optical science at the faculty of the Department of Solid and Crop Science here at Texas A&M. So Aart has been a professor at Texas A&M since 2019, and so he continues his groundbreaking work, and now you'll hear about his recent achievements from his talk. Aart,
thank you, Alexei, for those kind words. So my job title now has become ambassador, because I am your ambassador at Soil and Crop Sciences, and according to the people at Soil and Crop Sciences, I'm their ambassador here. So
you are double-edged.
Probably. Anyway, as Alexei was saying, right, I did a lot of laser development already during my PhD for those attosecond experiments, and then as a postdoc in Vienna, and that work got me into also into the field of multiphoton microscopy and optical coherence tomography. And some of the lasers that I helped to develop at that time, you will actually even see in the slides here, because we keep using exactly that laser. We could bring it from Vienna to here. So let's see how I get to the next slide. Not like this, like this. Good. you probably recognize a lot of those faces. So I will talk about a couple of different subjects, show a few different experiments that we're working on right now here. The first is related to multiphoton microscopy, where we do deep imaging deep into the sample and discovering actually some new things that are learning new things in Streptomyces bacteria. Then the second part is some development to help us learn even more things, so to improve the resolution of multiphoton microscopy. Then I'll change gears a little bit and talk about some other subjects, where the last two subjects have Raman spectroscopy, Raman imaging in common. First, we use it to learn some new things in plants, in roots, and then we even find out that we can use this Raman spectroscopy together with optical coherence tomography to predict whether seeds will be able to germinate or not. So I'll come to all the whys for these things when I get there. So I'll start with this deep imaging. If you've heard about multiphoton microscopy, a big advantage of using this multiphoton, this nonlinear effects, is that you get automatic sectioning of your sample. So when you go and focus deeper and deeper and deeper into your sample, you excite only things where you want to excite them deeper in the sample. So for that, you can use, for example, 2-photon microscopy. And then you would wonder, right, why do people want to go further and do, instead of 2-photon, 3-photon? Right at the bottom, you see 3-photon is a lot harder. It's harder to excite things with 3-photon absorption. You need higher peak powers. That is why we have this very nice laser. But the advantage that you get is that you can image even deeper and you get even better contrasts. So we'll go on a little bit. So the first thing that I will show here is this laser that we built, well, a decade ago, about in Vienna. This laser was not built for nonlinear microscopy originally. It was built to be the front end of a very big laser system for attosecond science, for attosecond experiments. We actually even used it for that. But at some point we thought, maybe this laser is good for more than just being the beginning of a very big laser. Maybe the laser itself has some utilities. And it turned out that the parameters of the laser, the repetition rate in the low but existing megahertz repetition rates, high pulse energy, it can be even higher, right? We managed to go with the same laser to a couple of microjoules with pretty short pulses for a fiber laser. With that kind of laser, you can do very nicely multiphoton microscopy and do very, very fast imaging, faster than that was possible with any other laser that was being sold or built at that time. Right now, you can buy some lasers that get similar specifications, but those lasers are still more complex than this one. Typically, to get into this regime where you get below 200 femtoseconds with a fiber laser, or with a laser that runs with this kind of energy, microjoules and 4 megahertz, you typically need an extra step behind your laser, and this is all just directly from one laser. Anyway.
Is it
So,
a long cavity, or how do you get
it's not a long cavity. Thank you for the question, Alexei. You start with a laser, you start with an oscillator, with a front, kind of always laser chain is often a chain of different components The first thing is the part that either generates the light or creates the pulses, right? So we start with an oscillator that has a repetition rate of a couple of tens of megahertz. Typical oscillators run around between kind of 50 and 100 megahertz. Then a couple of years ago, the Nobel Prize in Physics was given for a thing called chirped pulse amplification. That's what you need to do here too. So in the very first experiment for chirped pulse amplification, people took a femtosecond oscillator, sent the light into a very long stretch of optical fiber, making the pulses longer just by using the effect that the refractive index for different wavelengths is slightly different. So the speed of light for different colors is slightly different. That stretches the pulses, right? Different colors in a pulse go at a different speed. So after a kilometer of fiber, a pulse that can be 100 femtoseconds is suddenly a nanosecond in duration. So it gets stretched by a factor of 10 to the 4. What we then do is we throw out a lot of the pulses and transmit with this called AOM, fiber coupled AOM. We transmit one pulse every 250 nanoseconds. So we get about 4 megahertz repetition rate. And then we amplify those pulses. After you amplify them, because they are stretched in time, their peak intensity is lower, is not as high as that they would be if you wouldn't stretch them. So you do not get any damage at the end of the fiber. You can imagine, right? You amplify light pulses, and then you need to, as long as they're in a fiber, you could imagine things are manageable. But when you get to the output, to an interface where you get a jump in the refractive index, that's where you automatically generate nonlinear effects and you can get damage. But because we stretch those pulses, we do not get damage at the output. We need to compress them again. We use gratings for that, and then we send our pulses to the microscope. So when you do multiphoton microscopy, you absorb not one photon to generate fluorescence, but you absorb, for example, 2 photons. So our laser has a central wavelength of 1040 nanometers. So if I absorb 2 photons of 1040 nanometers, that would be the same energy as absorbing 1 photon of 520 nanometers. So kind of green light being absorbed. And then you can generate fluorescence of a longer wavelength. You would never, when you absorb 2 photons, you would never generate fluorescence of a shorter wavelength than 520 nanometers. When you absorb 3 photons, it would be as if you would have a 360 nanometer excitation. So you can generate blue fluorescence. So when we see blue fluorescence, we know that this cannot be due to 2 photon absorption. It could be 3 photon, 4 photons. The more photons, the less likely. So most likely that it's 3 photons. Okay. So, and this all is not possible with the type of laser that's typically being used for nonlinear microscopy, which is typically a titanium sapphire laser. They can be made tunable, and you can get a pretty high average power from this titanium sapphire laser, but you cannot get into the hundreds of microjoules. And 80 megahertz repetition rate, and that's kind of fixed there. You could, of course, use an AOM, right, to throw out a lot of pulses, but you do not get a high pulse energy for that. So this laser is kind of unique for being able to do this. So now what we want to do with that, right, when we do this 3-photon deep imaging, we developed it for monitoring in vivo natural occurring compounds, if we use autofluorescence or other processes that you can study by adding in some genetically encoded fluorophores. But in this case, when we study Streptomyces bacteria, we want to look at their natural occurring compounds. Some of them can be antibiotics or anti-tumor drugs, so that can be very relevant. These kind of samples are very highly scattering, so that's why you want to use also this nonlinear microscopy with single photon absorption. You would basically just see this instead of nicer colors with more detail, and on some next slides you will see even better details. Here you see a couple of those Streptomyces filaments. They're kind of filamentous bacteria. And we look at the fluorescence coming in different color channels. So if you see some red here, it is in this channel of 470 to 640 nanometers. That's 2 photon excited fluorescence. We have a measurement channel that spans from 490 to 560 nanometer. As you remember, 520 nanometer is the threshold from where you can get 2 photon fluorescence. So below 520 nanometer cannot be 2 photon. So you see some green here, right? Sometimes they overlap, and if all colors overlap, you get something white, of course. And then we have the blue fluorescence, which is 3 photon. So for this measurement, we took the same image and varied the power from the laser, and just try to see what is the power dependence of the signal. So for the red signal, the power dependence has a power of 2, which means it's 2 photon fluorescence. For the green, it is 2 to the power, or the laser power to the power of 2.8, which means that it's kind of a combination of 2 photon and 3 photon. There might be different fluorophores, some that are 2 photon, some that are 3 photon, and they are just adding together. And then the blue signal scales to the 3rd power of the laser intensity. So that is obviously 3 photon excited. And as I said, right, it's the most important part of this nonlinear imaging is being able to look actually not just at the surface of the sample or slightly below the surface, but deeper into the sample. This sample had a attenuation length of about 80 micrometers, which means that after 80 micrometers, you get left with about 30% of the laser power. And 80 micrometers deeper, 0.3 to the 2nd power, so you get much less, right? So you could imagine that if you go deeper and deeper and deeper, you start losing signal. You can increase your laser power a little bit to accommodate for that. But when you have a 2 photon absorption or a 2 photon fluorescence, increasing the laser power means that you have more power at the top of your sample, and that can also start to generate this 2 photon effect. So with the 2 photon excited fluorescence, you start to get kind of a little bit of a blurring or a masking of your signal with the background. Whereas with the 3 photon absorption, needing 3 photons to generate signal, you do not get as much background from other layers than the focus where you're looking. So when we start to quantify that, you can see, right, the signal to background, basically the out of focus fluorescence that you generate increases and increases as a function so the signal to background decreases if you go deeper and deeper and deeper. So after even after about 150 micrometers, 2 scattering lengths, you get as much signal from outside of the focus compared to what you get from the focus. So, right, you can still see a little bit of changes in the contrast, but not very much, not very well. Whereas for 3 photon, it stays basically almost constant. And when we go even deeper at some point, the it kind of jumped from a signal to noise of 10 to 100. Sometimes the density of the sample changes a little bit. So, yeah. Right. And then when we go even deeper, so this is at a depth of like 780 micrometers, so almost a millimeter below the surface of the sample. That is, in this case, for a sample with 120 microns attenuation length, more than 6 attenuation lengths below the surface. In the blue channel, we can still tell apart all those filaments, whereas in the red channel, even when we adjust this, the contrast of the image to be most favorable to view it, it's just all blurred. So actually the three photon microscopy allows you to look a lot deeper than with 2-photon.
So Art, I had a question.
Yes.
For 2-photon case, if you increase the power, would it help to get better signal to noise?
No, because right, if you increase the power linearly, right, well, you get like the signal squares, right? But on the surface, you also increase the power in that same way. So you also start to get a square dependence of any out of focus fluorescence. So they just scale up together. Now, you might start to wonder, right, what is actually this blue signal? I couldn't find the nicest pictures here. The previous pictures, I helped your eyes a little bit by smoothing them out a little bit. And these are without any smoothing, but you see these a little bit of the blue signals over here, right? So this is all autofluorescence. The blue is in this wavelength range, the green in this, the red there. For this wild type Streptomyces bacteria, we get some of this blue fluorescence. Now, our collaborators were very interested in actually the linear mycin that those wild type bacteria generate. So they had created a mutant that doesn't create it, just to see what the effect is, right? If it's some kind of like anti-fungal agent that would kill fungi or other bacteria. So in this mutant, when we put that mutant under our microscope, there was no blue signal at all. So it turns out that this linear mycin is actually what causes that blue fluorescence. So we can use this blue autofluorescence to find out things about where are these bacteria producing it, when are they producing it, and how other functions in the cells or in the bacteria. So in a follow-up experiment, we looked into the how this blue fluorescence is being generated. So in this case, the red color is a membrane stain that we added to the sample. So you can see kind of sometimes like these kind of thin lines, right, that are demarking the membrane, the outer walls of the bacteria. And you can see where the blue fluorescence is happening. And again, in the mutant, there is no blue fluorescence. There's a little bit of kind of dirt somewhere over there that created a blue signal. But we can use these kind of things together to find out so this linear mycin is often trafficked in vesicles, kind of little lipid pockets, like RNA vaccines, right? And by using different membrane stains, you can then find out what kind of lipids are encapsulating this linear mycin and how are the bacteria actually changing themselves if there is linear mycin there or not. Okay, so now we'll go over to the next one, right? You can imagine if we're looking here for these kind of like much narrower structures, right? We start to get to a point where the resolution of your microscope is not enough. So that's kind of the natural reason why you would want to go for improving the resolution of your nonlinear microscope. The resolution is typically known for a long time as typically like the wavelength divided by 2. Sometimes if you have a very good microscope objective, it can be a little bit more, a little bit better. But right, if you would start with green light, 500 nanometer, you get a resolution of about 250, 200 nanometer at best. So if you want to get better, you need to go beyond that, which is called super resolution, which also got a Nobel Prize 10 years ago. So when we do laser scanning microscopy, what really matters is the spot size of your laser. So if we take that, we get like 0.4 times lambda divided by the numeric aperture as the radius, the 1 over e square radius of the light. For full width half maximum, you would get about half of the wavelength again. Now, when we do take nonlinear effects, right, we take basically the spot size of the laser and take it to the 2nd power or to the 3rd power, which would mean that the width is reduced for 2 photon by a factor of square root of 2, and for 3 photon by a factor of square root of 3. You might think that is good news, right? But it's not 100% good news because, of course, we take a lot for nonlinear microscopy for multiphoton. We take a longer wavelength, right? So for the same fluorophore, we need 3 times the wavelength or 2 times the wavelength. So if you get actually a worse resolution with multiphoton microscopy than with normal microscopy. So to go to super resolution with multiphoton microscopy is even more important. So how do we do that? We use something called an array detector with a lot of small detectors together, in this case 23, that are all smaller than the diffraction limited resolution of the microscope. Right? So they're, of course, a couple of micrometers in size, but because the microscope does this magnification of the image, but they're smaller than what a diffraction limited spot would cover. Now, when we use that sensor, normally with nonlinear microscopy, you put your sensor, your detector, as close as possible to your sample. For this image scanning microscopy, we need to go all the way back and route it back over the scanning mirrors and then come onto the detector as if it would be a confocal microscope, actually. Then, right, for example, with 3 photon microscopy, this would be your laser spot size. If you would say the fluorescence light that you generate would give you a detection point spread function resolution of about this. But then if you put those 23 detectors together, you can imagine each one of those 23 elements would have its kind of optimal imaging point slightly shifted away from the maximum of your excitation function, right? So it would be kind of a detection that is slightly to the side. So if you want then to calculate what your signal is, you would just need to multiply these two together, where this one is shifted for each one of the detectors. And by then, in post-processing, shifting all the different images after laser scanning for each of those 23 detectors, you get an image that has a better resolution. So it's easier to see than the results, right? Standard microscopy would give you like big blobs. They're still pretty small, right? The full width half maximum of this blob is 300 nanometer. when we use the image scanning microscopy, the size of this blob changes already to 190 nanometer. So that's a factor of, it's about 60% improvement. From here to here, it's 1.6. And then you can use something extra, which is called deconvolution, to improve it even better. And you can get to a resolution that is about 126 nanometer. So a lot better than what you can even get with when you would start with 400 nanometer light and detect this in this blue detection window. Right. And you might think like, well, but you can only make very small images, right? This is, if this is 300 nanometer wide, the full width of maximum here, then this is maybe 2 micrometers. That's not looking at very big things, right? But we can actually take an even larger field of view. So this would be maybe 20 micrometers, 30 micrometers. And then we can get resolve a lot of different structures of a size of 100, 120 micrometers in that, or nanometers in that 20 to 30 micrometer size. Then the advantage, again, is when we use 3 photon excitation, we can go from the top of the sample to pretty much deep down and still get a good signal and good resolution. So in this case, the objective that we use has a working distance of a bit more than 100 micrometers. So basically, when the objective is really touching the sample, you look about 100 micrometers below the surface, and we can get, with this image scanning microscopy, we can resolve still better images than when we use only what is called non-descant detection, when we use the detector over here. So there is a big advantage there. Well, we can, of course, put several of these detectors with different color filters in front and detect these different colors, all with a sub-diffraction limited resolution. So we can do 2 photon and 3 photon image scanning microscopy all at the same time, and get an improved resolution over when you do standard 2 photon and 3 photon microscopy. We can improve it even more by actually adding something that is called super resolution optical fluctuation imaging. This was the idea of a former postdoc from Girish that then worked with us for some time. So I will just show you what this optical fluctuation imaging is. When you take a series of images from a sample, you often get like a little bit of blinking of different fluorophores. So let's see if I can play that again, right? You see all those kind of different dots that are all quantum dots in this case. They kind of sometimes fluoresce, sometimes do not fluoresce. And that kind of statistical blinking you can use to your advantage to improve the resolution by taking kind of a correlation, a time correlation of your signals, of your image signals. So you can see here, right, these different dots, they all switched on and off at some points. So we can use that. When you use this second order correlation of the time series of the image, you improve the resolution by a factor of square root of 2. So we can actually combine that with this image scanning microscopy. So this is a normal photomultiplier measured image of, in this case here, we see 2 quantum dots. We can see that there are 2, right? But we cannot see if there is something maybe in between there because they're kind of merged together. And when we do only image scanning microscopy, we already kind of see that there is a real good minimum in between. When we add these fluctuations to the analysis, then we can see that they get even better separated, right? The center of mass, of course, stays at the same position. but so when we take the line out there, you can see that with the photomultiplier, there is a slight minimum in between, but they're really merged. With the image scanning microscopy, which is the blue trace, there is a very well-defined minimum in between, but it doesn't go exactly to zero. So you cannot still not tell whether there is really nothing in between or there is maybe something very faint in between. And then when we use the image scanning microscopy and the SOFI, then we can define that really it goes to zero in between the two elements. So we can resolve things a lot better.
The red is without the fluctuation analysis?
The red is without fluctuation analysis. If you would do only you could do, for example, fluctuation analysis on this image too. Then you would improve it to about the blue trace. But the two techniques together improve it by a factor, well, almost a bit more than a factor of 2 together. Each of them a factor square root of 2 or slightly better. So with that, we got to a resolution of about 100 nanometers. In this case, by using 2-photon imaging with the 1040 nanometer. So we would get to about, with the ISM, we would get to about again, to about this 190 nanometer, maybe at the very best, and then to about 100 nanometer. Well, okay, so let's go to the next experiment. We started to look into nitrate in plants. Of course, as you know, nitrate is one of the most important nutrients for plants. There is basically, right, you would, if you know about fertilizers, you might know them by an abbreviation of NPK, nitrogen, phosphorus, and potassium. Those are the three most important nutrients, and nitrate is one of the ones that is needed in the largest quantity. And it has a lot of functions for plants. Signaling, it's of course needed for growth. And the way it's taken up from plants is actually all through the roots, right? Nitrogen, there's a few plants that can take nitrogen from the air through the leaves, but those are real few exceptions. So all those nutrients are taken up with the roots. So we want to start looking at how roots do take up those nitrates, those nutrients. And the most fundamental aspects of that nutrient uptake are actually not so well understood. You can imagine it's hard to study actually roots because they're in the soil. Makes them hard to do kind of microscopy imaging with them. Most of the studies are actually done by kind of manipulating different genes. And in all these kind of nutrient uptakes, there is a lot of different genetics that are taking place there. But there is a lack of methods for a good visualization of the spatiotemporal characteristics of this. So one of my colleagues in the Department of Soil and Crop Sciences is actually studying nitrogen management in plants as her major field of study. So she asked me at some point, would we be able to detect nitrate specifically in roots? And the concentrations that you would expect in roots or in soil for roots to be taken up is typically less than 10 millimolar. Well, that doesn't say very much to any of us, right? But this is kind of a starting point for a concentration that we would need to be able to detect, or better than that we would need to detect. And of course, we want that kind of non-destructive, right? So we started with this very nice microscope that we have down here in the basement. We need to collect enough signal to detect below this 10 millimolar, which is kind of the physiologically relevant condition. But we don't want to burn the sample. Right, when you do Raman spectroscopy, you shine your laser light on your root. If everything is good, you see this. If something is not good, you see this. And that can happen very quickly. In Raman imaging and Raman microscopy, this is a big problem. You very easily damage your samples. And another problem is fluorescence background. So, right, we started this with this Horiba Labram microscope. We got problems like the signal was extremely low, and we noticed that actually our imaging was kind of like, it looked like we were losing our focus over the time that we were trying to measure. And that can have different reasons, mechanical stability, or even water evaporation, right? We were just having roots there, plant tissue. If you leave it out in the open, it will dry up and shrink. So all these kind of things can happen. So we got then a new microscope here, a new Raman spectrometer that is coupled with our microscope that we built here. We put our plant right under the objective. Actually, we have our seedlings grown on these kind of plates, plastic plates with some aqua gel agarose on there, and then the seeds are just dropped here in a line. We leave this plate standing vertically, and the roots start the seeds start growing, and the roots grow nicely straight down. Roots like to grow in the direction of where gravity pulls them. So you need to keep them standing up like that. Right. If you look at the microscope, you would see like the roots sitting here with a little bit of a black shadow around it. That is some deformation of the agar gel. Further up the roots, you see here, these are root hairs. Those are the things that roots actually use to take up nutrients from their surroundings. And then when we take a Raman spectrum, the black, can you see it black? Yeah, you can still see it, right? You get a black trace, you get a lot of peaks. When we get a spectrum, the blue spectrum is just the agar plates, all right? We just, for the black one, we focus here. For the blue one, we focus somewhere far away where there is no root. And we can see some of the peaks are actually coinciding. But for example, this peak is only there when the root is there. Somewhere over here, something here. So we can kind of isolate a Raman signature from the root and tell it apart from the agar plate. If we take the root alone for a very short time, it will survive a little bit. Then all those big blue peaks are gone, and we get left with most prominently actually the peak that corresponds to the nitrate. Right. So for that, we needed to do a lot of work. Actually, we needed to control the hardness of this growth medium. If it's too soft, the roots will start growing inside sometimes. And if it's too hard, you can imagine the plants could just fall off. We needed to keep them vertical in the growth chamber so that the roots really grow straight down. And we need to keep, of course, all parameters the same, except for the nitrate if we want to look at what the effect of nitrate is. So on each plate there are 15 to 20 seedlings. We measure typically about 5, sometimes a bit more per plate. And then we repeat the measurement a couple of times. think the smallest repeat has been 4, and sometimes with some concentrations we did more than 20 times. So we measured tens, hundreds of roots for all this. So you could imagine, is that worth all that, right? Well, we come to that. First, can we actually quantify how much nitrate we have? So when we have on the agarose, on the agar gel, we add nitrate to it. This peak of nitrate grows linearly with the concentration that we put in there. So we can actually, from the peak height of the signal, extract what is the concentration of nitrate. And then we hope that extrapolating that calibration to the roots is valid. But one thing we know, right, if it's in a root, it will be also linear. So we might be off by a constant factor, but we would be off by the same factor for every root. And well, from this, we can say that we can quantify the nitrate on those plates down to a concentration of about 1 millimolar. So that's about our sensitivity. Better than 10 millimolar, so good enough. So now we started measuring the nitrate concentration here at the tip, then slightly away, further away along this root. And what we found is that the concentration of nitrate in the root always grows up to kind of a certain plateau in the root. And so what it turns out is that these first 3 millimeters of the root are the part where the root is still growing, where cells are elongating, new cells are being formed. And after 3 millimeters, the cells are basically in their final state. They have elongated, they have formed their cell wall, they cannot not change anymore. They're still alive, but they cannot change their size anymore. And it is in those mature cells that the nitrate concentration is highest. And what we believe, what is believed is that the nitrate is actually stored in the vacuoles of those plant cells. So after a cell has been matured and has formed this vacuole completely, that is where it would start accumulating, storing the nutrients. And then where the cells are growing, it's actually where the nutrients are, of course, being used too, right? So if they're being used, the concentration gets lowered just by the use. Right. So now, of course, we wanted to check whether that was really true, right? So we took another method, so something called the salicylic acid method, that needs hundreds of seedlings per treatment, right? For each of those, say, for example, for this 10 plants grown at 10 millimolar, that's maybe 50 measurements, 50 roots. For a validation measurement, we needed 500 roots. So you need a lot more. They're cut in pieces of 3 millimeter, first from 0 to 3, and then from 3 to 6 millimeters. And that shows that we get higher nitrate content in this second piece of 3 millimeter. You can imagine, right? 3 millimeters is cut off here. We get, with the Raman spectroscopy, we get a resolution that is a lot better. We get far beyond that 3 millimeter resolution. So what you can see here, let's just look at the rightmost one. The first 3 millimeter has a concentration about, well, a little bit more than a third of the concentration of the second part of 3 millimeters, which is more or less the same as what we saw in our Raman measurements. The numbers, however, are off by quite a bit. We measured something like, at the highest concentration, maybe 10 millimolar inside the plant. And then according to this measurement, it's about 40 millimolar. So maybe this light scattering in the root causes us to lose like a factor of 4 of the signal. But in any case, the ratio is correct, right? So that's another confirmation that we can use this Raman imaging, Raman spectroscopy to quantify how much nitrate we have in our plants. Another thing that is interesting, actually, when you go to even higher concentrations in the growth medium, the nitrate concentration in the roots doesn't grow anymore. So when there is a little bit of nitrate, plants will take it and collect for themselves to store. If there is a lot of nitrate, if there's too much, plants actually say, stop, stop. So it's what you can see from this is that it is not something like diffusion of nitrate coming into the plants and plants being happy with it getting. Plants actively search for it and get it in and concentrate it. So, okay, I'll speed up really, really quick. When you have seeds, and for example, cotton seeds, we did a measurement and checked how many seeds germinated, grow into a cotton plant after you plant them. For this variety here, out of every 100 seeds, you get 40 plants. If you want to do precision agriculture, you're in trouble, right? Because you plant 100 seeds or 1,000 seeds in a field, or 1,000,000 seeds, and you get just 40% of that growing out. That means that you get a statistical distribution of how they are. Some patches are very dense, some patches have nothing. For precision agriculture, you like your plants being spaced out evenly. Every plant have the same amount of nutrients available, the same amount of space, the same amount of light. So this is not good, right? We would want to actually be close to 100% maybe. For this, it's already a little bit better. For this, these are very fresh seeds. They're quite good, not super good, but okay. So can we do phenotyping to the seeds to tell whether they are germinating, whether they're viable? Again, you can have this problem, of course, right, when you do Raman spectroscopy. So we needed to be careful. Actually, we needed to check them at like a couple of different places to find out some useful information. So when you take Raman spectra at these 10 different positions, you get something like this with this fluorescence background and everything. So we needed to normalize them. And actually what I show here is the red here is showing the standard deviation of on the average over here. So this, where you see a lot of red, that means that the spectrum is actually at different places, especially in this region, is very different. The signal is very different. Whereas other places, of course, we normalize to this one, but at other places, these peaks are kind of very stable and in height, in strength. So maybe this kind of variability is something, right? The second thing that we do is optical coherence tomography. That gives you some kind of morphology of your seeds. So if you scan through, in this case, it's like if you were scanning, you look from top to bottom, and you're getting like a height profile of the seed while you're processing along the seed. As you might have noticed, sometimes there was some scattering, some light, some signal much below this kind of the hole that you see here, right? Let me see if I can repeat it once more, if it finishes. Right, you see it there, and this is kind of a deep trench in the seed. Well, what do you think is actually the problem with the seed? The seed is just damaged. A cotton seed, the cotton fibers grow on it. They need to be pulled off, right? We grow cotton to make clothes out of it. We want the fiber. But when you take the fiber off from the seed, sometimes you damage the seed. That's a problem for the fiber quality and for the seed. The seed is not viable anymore. It's damaged. and the fiber was also not as good quality. So, but if you'd be able to catch this kind of damages that are not visible to the naked eye with the technology, you can tell, okay, this seed will throw away. So we got OCT scans from different seeds. This was the seed that I showed here, the image of, with this kind of very deep pinch inside. Another seed was looking like it had a little bit of a fuzzy outer shell, whereas this seed here, you see kind of two different layers in the shell, in the coating of the seed. So we see also kind of differences between individual seeds here, apart from the damage. So now when we lined it out with growing up those seeds just after measuring them, damaged seed, this seed might have been doomed from the beginning because it also had a very high spatial variability of the Raman signal. This seed with this kind of fuzzy Hall signal also had this very high variability in the same spectral region. And this seed that actually grew up was having a very stable, homogeneous Raman signal throughout the seed. So by combining the OCT and the Raman, we can then start to try and make a prediction whether a seed will grow out or not. So the ones that we predicted to germinate for this variety, well, it got a little bit better, right? It got from 40% to 50%. Nowhere close where you want it. You would want it maybe 99%. For this variety, it went from below, just a little bit below 70% to almost 90%, which is actually pretty good then. And then for the last variety, it still improved. What you see here in between is actually, we also checked just all the seeds that we measured, right? And controls are seed that were not measured with Raman or OCT in any form or fashion. After you've been measuring and handling them, it seems that the germination rate goes up a little bit. You could think that maybe there is some kind of radiation therapy done to the seeds too. But the prediction still beats that. We can show it slightly different, but I will just skip over that. One thing that we can think that might still help there is to add some machine learning to this, to maybe catch some more fine details in the spectra or in the OCT tomograms. So in summary, I told you about multiphoton microscopy and super resolution with that. I told you about the Raman imaging in roots, where we see that roots in mature cells take up nitrate, concentrate it, or limit it if the concentration is too high in the environment. And there's a lot of things that we can kind of follow up on that with having this tool available to us now. And with that, I will leave you and give you the opportunity to do actual work.
Thank you very much, Aart.
Just a quick question. I want to make sure I understood correctly. Exciting work, by the way. When you find a damaged seed, do you try to plant it and see if it grows, and it never does? Is that the case?
Yes. Yeah. We still tried because we're stubborn, but yeah, we could have just thrown it away. Then one.
Yes, Raman.
I'd like to know, like, who made the prediction? Like, or what's the actual protocol criteria?
So we had two types of measurements, right? We had the OCT and we had the Raman. If we saw this in the OCT and we saw high spatial variability in the Raman signal, then we predicted them to not germinate.
Just individually, looking one by one.
Yeah, I mean, you can extract a number from this, and so you can have the computer do it, but
Artificial.
well, even artificial stupidity, just right, you extract this variability if it's above a threshold, and you've also labeled this as being fuzzy, then you put a label no. And for 300 seeds, you can write a script for that. Artificial stupidity.
I suggest at this point, we thank Aart once again for